Good afternoon. My name is George Paris. I'm a PhD chemist who got interested in biology about 20 years ago. And in particular, I've been working on the question of how does a cell, single cell, that we all started from, how does that differentiate and develop into something like a human being or any advanced animal? When I started looking at the different hypotheses for how that happens, I noticed that most people seem to be talking about gradients, chemical gradients, etc. Wasn't really impressed by that. I'm a chemist and I know that chemical gradients are not necessarily very stable. They vary over time and space. And so I really didn't like that idea at all, have trashed it in several situations. And I started developing my own ideas. Now, what I've done in the last, I think I've done about 4 videos before this one, in which I've developed different elements of my ideas about development and evolution, all of which are probably pure heresy for the biology community. I have the advantage of having no biology experience past high school, basically. So I have no parochial interest in maintaining the sanctity of biology and its theories, or physics for that matter. I'm a chemist. Um, and what I have done is I have developed these ideas and they're hypotheses. I want to encourage you to understand that they are hypotheses. As a matter of fact, everything we think of as a fact is really hypothesis. You know, from the 60s, Bernard Shaw's like never resist temptation, test all things, hold fast those that are true. So I'm that kind of guy. I'm a 60s kind of guy. And so hypotheses are not truth. Hypotheses are a tool for finding truth. And all I'm doing here is trying to give you a tool, those of you who are interested in this topic, to maybe find truth through experimentation and whatnot. I do not rest my ego in these ideas. I rest my ego in the process of answering these questions scientifically. Okay? When you do that, by the way, it frees you from, I can't let this idea die because it's mine. I'm tied to it. No, I'm not tied to it. I am basically waiting for somebody to prove me wrong so I can change it. Okay? So if you can prove me wrong, I'd love to hear that I'm wrong. I'd love to hear that I'm totally screwed up. That's the essence of science. That's where I want to be. Okay? Because if you tie yourself to a particular idea, it's going to change because science is always discovering something new. All right. Now, those of you who do get interested in what I'm saying, what I've been talking about in these other videos, I have published a number of papers. You can find, if you search Paris GE, that's my initials, George E, on pubmed.gov, you can find papers I've published. If you can also find them, I'm sure, in Chem Abstracts. I've done some chemistry papers as well. And then a few years ago, I got excited about Amazon's self-publishing capabilities. What I'm going to talk about today is actually primarily in this book. There's several other books I've published, and there's also, this is a paperback version. There's also a Kindle version, which is earlier and more elaborate, more detail for those of you who want some background details. This area is moving very quickly. 20 years ago, nobody believed in non-coding RNA. Now, everybody believes in non-coding RNA. So this is where we're going with this. This book, it's 100 pages. The last 30 pages are basically references to the scientific literature. So I'm not winging this entirely. I look into the scientific literature for ideas and for collaboration, for confirmation, and for situations where I can't explain it. What I haven't done so far is I have not found a situation where this hypothesis is flatly contradicted. Basically, I reinterpret some of the scientific findings in context here, but I'm not in contrast with any particular finding as far as I'm aware. If I am, let me know. I love it. Okay, so the name of this particular activity is what I call the Master Developmental Program hypothesis. Master Developmental Program hypothesis. I started out looking at the issue of development. How does this single cell develop into a complex human being? And I found out originally that people were talking about gradients. As a chemist, I didn't believe that. I would modify that to the following extent. If you look at plants, plants do respond to gradients of moisture, temperature, sunlight, probably gravity. Okay? So plants do, trees do follow gradients. But if you look at a plant, if you go to an encyclopedia of plants and you want to know about different trees and what the anatomy of an oak tree or anatomy of a willow tree, what you will find is a description, a general description. This tree grows tall and straight, it grows short and fat, it branches weep out, whatever. You get these general descriptions which are consistent with gradients, but you do not get a picture of this is how an oak tree branches. It's not, every oak tree is different. They respond to the sun, they respond to whatever happens around them, and they branch accordingly. It's sort of random. It's basically driven by gradients of some sort. It's not programmed the way that a human being or a giraffe or whatever centipede is programmed. There's a program driving the development of basically of animals. I see that's the big divide between plants and animals. Plants do respond in gradient form, whereas animals have this built-in program that allows them to go into these exotic different shapes, and we'll talk about that along the way. Now, the key to this whole process, and the part that I think was probably my most novel idea, had to do with how cells differentiate. Now, if you look into asymmetric division in the current literature, you'll find lots of talk about polarization of the cytoplasm and whether it's caused by polarization outside the cell or inside the cell. I think that's kind of nonsense, frankly. These papers that talk about asymmetric division almost never explain anything about what's happening in the chromosomes and the nucleus. They all talk about what they can see through a microscope. This is one of the hallmarks of biology. They like to talk about what they can see through the microscope, and they can't see the chromosomes. They cannot see how the actual molecules are moving, although we're getting better at that area, but we're not there yet. But basically, biologists are driven by what they see through the microscope. Now, I take an entirely different view of that. And what I talked about in my most recent video, which you may or may not have seen in our correct order, had to do with asymmetric division and something called the immortal strand hypothesis. All right. Well, basically, let me go into this. If we start looking at this process, and I don't to distract you too much by this, let's look at this. If we start off with a cell, I have it upside down. That's a problem. That would be the problem. Yes, that's why it didn't look normal. So, okay, I can't see it as well as you can see it, hopefully. In any event, I do this in my basement, people. This is not done for profit. I'm talking to you the way I would talk to a college class if I were teaching this in a college. Nobody's going to let me talk about biology in a college class, but I do teach chemistry occasionally to college students, and this is more or less the way I would lecture to them. But this is not a for-pay process, and so I'm doing this in my basement. If a cat walks through, it's my cat. Don't worry about it. Okay, so if my phone rings, I might have to cut that off. But anyway, you'll hear me do some rude things. So anyway, here's where we start. You have a cell, and I use these, it's actually the @ sign, to make up the strands of the DNA, and then these X's I use to represent the epigenetic marks on the DNA. And you'll notice in the original cell, the epigenetic marks of both sides are the same. And when you go through a cell cycle and you get to the G2 phase, You've got the epigenetic marks on the original strand, but you do not have epigenetic marks on the other strand, the newly synthesized strand. By the way, the original strands are just regular dark print, and I've highlighted all the new material that goes through here. Somebody's trying to hit me on Facebook. Who cares? I don't watch Facebook much. Let me get rid of that. Boom. I hope. Nope, doesn't want to go away. Okay. Hope you can't see that. In any event, it's very persistent. There it goes. Finally, it's off the screen. I hate that. Don't want to be bothered. In any event, here we have the original strands, the new strand that does not have epigenetic marks. Then we go down here and we go into G2. And my hypothesis begins with the idea that somehow in this G2 phase, this is obviously a stem cell of some sort because it's differentiating. Somehow in this G2 phase, there is a guide which causes the new strand of DNA to be marked differentially. In other words, it doesn't have the same epigenetic marks as the original strands, okay? So now you have this situation where you got these cells where one strand of DNA is marked one way and the other is marked the other way. And this, I believe, is where you start getting polarization of the DNA. All right? Now, don't worry about this for a moment. I'll get back to that. So this is where you should, as a skeptical student, should start saying, well, where does this come from and what's its format? And I believe that the marking of the DNA in the new strand is guided by non-coding RNAs. I think there's some evidence for that. And so you're basically non-coding RNAs come in here and cause this differentiation in the coding of that is to say, the epigenetic marking of the DNA. Now, when you go through the next cell cycle, and this is interesting, when you go through the next cell cycle, if it's a normal cell cycle, then you end up with a situation where one of the cells, one of the strands, one of the new old strands of DNA becomes a new strand, okay? And the original, the immortal strand, produces a strand identical to itself. So now this cell is the identical to the original stem cell. It doesn't have to be told it's a stem cell. It doesn't have to know it's a stem cell because where it's located or anything else. It knows it's a stem cell because it's got the right markings for that cell. The new cell, on the other hand, has an entirely different set of markings. It is a new cell. It is going to differentiate differently. It's going to behave differently. It's going to have a different proteasome. Okay? So the immortal strand follows through and stays with the original stem cell, whereas the new cells have completely different epigenetic markings. Okay? Now I've had to introduce some terms, and I've put them over here. I think I can do it this way. Get closer, let you see it. I'm using the term, something I call the generation-specific control key. And that's a string of non-coding RNA generated specifically in one generation, and it ultimately determines what happens in this process. Now, the generation-specific control key it's just a long strand. It's got lots of different pieces, and those pieces I call nuclear messenger RNAs. In other words, the control key is chopped into a bunch of pieces, which I call nuclear messenger RNAs, and they fan out into the DNA, the chromosome, the genome, and they cause these epigenetic markings to occur. Now, why is it done like this way? And I created another term called the delocalized operon. And let's go back to where that term comes from. If you look at bacteria, nice circular DNA, if you're going to have a biochemical process, some substrates converted into some final product, You typically are going to need several different enzymes to do that. Those enzymes each will be coded by a particular gene. And to make this whole thing work, you have to have all those enzymes present at the same time in the right process, the right quantities to make this process work. If you don't, you're going to have some sort of jam up in the biochemical processes and the cell probably will die. Okay, so what nature has done, that is to say, the things that have been selected for through natural selection, is that those genes have been lumped together back to back to back on this circular chromosome, and it has a start when a particular biochemical signal occurs within the cytoplasm. Remember, there's no nucleus, there's just a cytoplasm. It causes the transcription of all of those genes concurrently. We call that an operon. And then they're cleaved and they go out and do their job. So in a higher organism where you have, this is the eukaryotes, where you have a nucleus and where you have linear DNA, and we have very complicated processes that occur, different cell lines will have completely different biochemistries. And in order to make those biochemistries work, there are many different genes that have to be activated. And so you can't just line these things up and transcribe them all at once. They're spread, the different genes that have to work together are spread all over the different chromosomes. They're delocalized. It's an operon, but it's delocalized. And so these nuclear messenger RNAs fan out, find the right genes, activate those genes by changing the epigenetic markings. And so your next generation of the cell will have a completely different proteasome as well. I mean, the proteasome is based on a activated genome. It will have different epigenetic markings, and hence it will go forth and do different things. That's what we call differentiation. So what I just showed you was how one cell could end up being two different cells. And I think that's a fairly important thing, and it focuses, my focus is entirely on the genome, not on the cytoplasm, not on what's next door, not on any chemical gradient, but purely on what's happening in the chromosomes, the genome. All right. So that's the basic step. That's the most important biochemical transformation. That's how you get from one cell to two different cells. Now, on a developmental scale, this has to happen in many different ways on many different cells. Hope I don't lose my voice. If I do, I'll take a sip of my favorite soda. And hope that works through things. Loosen up the flame, you know. In any event, this whole process then depends upon what I call this generation-specific control key. And what you should be jumping up and down saying right now, where is it, and what is it, and how does it work, and most importantly, how does it evolve? One of the most rewarding things that, to me, from my thought process in developing this hypothesis of development was that when I got finished, and I'll explain this, you'll see this later, when I got finished, I could explain how evolution occurs. Evolution has very little to do with changes in the proteins. I think most people are here again, biologists are focused on the proteins. It's like, oh, we got some microscopic change in the protein, and so magically some tremendous change in development occurs. It turns out the proteins have almost nothing to do with evolution. We have pretty much the same proteins. All that biochemistry is the same in us and in bacteria and in every other mammal. All the mammals are very similar, and certainly humans and primates are all similar. So there's very little change in the proteins per se that change have any effect on evolution. Now, people will point to, I'll diverse here a second. People will point to things like albinism. Okay, sure. If you knock out a gene that is responsible for skin coloration, you will get no skin coloration. I mean, it's like, okay, that's not evolution. That's a some sort of a genetic transformation. It's not an evolution in any particular direction. It's just something that happens. So what happens to determine that I'm sitting here with 10 fingers, 5 on each hand, and my anatomy the way it is has very little to do with what happens to individual proteins. It has a lot to do with how those proteins are orchestrated. It's the difference between a guy playing playing a xylophone and an orchestra. Okay, there's a conductor out there making the whole orchestra work together, and that's what the developmental program is. The master developmental program is. All right, that's what I envision it as being. So, going back, the questions are, well, how is its construction? What does it look like? Where is it? And how does it work? Well, we do have some evidence for generation-specific activities. For example, we know that the telomeres are incremented generation by generation. For biochemical reasons, you can't transcribe or you can't replicate that end of the telomere, and so it gets cut back every time. We know this for example, in the original cloning experiments with the sheep. Okay, they took a cell from a mature sheep, cloned a new sheep, and the new sheep evolved just like developed just like the old sheep. Okay, there was no change in the development, but when that sheep was mature and you looked at the chromosomes, you looked at the telomeres, you found that it had shorter telomeres because that cell that had come from the adult that had become the parent of the clone had been shorter. So this tells us two things. One, there are cyclic processes going on, generation by generation, and that's one. And two, the generation-specific control key cannot be in the telomeres because they got lost, but the development still happened normally. Okay, so the question is, where would it be? Going back to the early 2000s, they tell you that we have sequenced the entire genome. That's not entirely true. Okay, the methods that were being used to sequence the genome work well for the euchromatin, but don't work well for the heterochromatin. The heterochromatin is too redundant, too many overlaps to make it work. You can't piece these pieces together. So the heterochromatin is still largely a mystery to us, and primarily the area of heterochromatins is what we call the paracentromeric regions. On either side of the centromere, there's a stack of this highly coiled DNA heterochromatin. Now, for a long time, people didn't think it was even transcribed. There was a lot, if you go back 15, 20 years in the literature, oh, it's not transcribed. Well, it has been shown that it's pretty much everything is transcribed. Okay, virtually everything in the cell at one time or other is transcribed. Okay, and the heterochromatin is transcribed, and it's transcribed in specific time frames which are consistent with what I'm about to say. In any event, let's think about this heterochromatin a minute. It has a very important mechanical function. When the chromosomes are pulled apart in the cytoplasm, there's a drag on them. You see it from the way that the centromeres are pulled out and the tails come back. It's harder to pull them, and there's going to be more tension in the rope, so to speak, at the paracentromeric area. And so it's good that you have this doubled up rope, this doubled up coil there, so that it's strong enough to pull everything apart. But we know that there are breaks in this nonetheless. For example, in humans, chromosome 9 is known to have paracentromeric inversions. Basically, the centromere popped out. There was a break on both sides of the centromere. It flipped over and came back in. And actually about 1% of the people listening to this video, assuming you're sort of a normal distribution of human beings, have inverted paracentric heterochromatin. Okay. By the way, this does not change your morphology. It does, however, have an impact on reproductive success. And I've talked about this earlier in my video that has to do with hopeful monster. Okay, I believe that this is the basis of the hopeful monster phenomena. This is how you can have a saltation, that is to say, one generation change, which will create a potential whole new species, and it also creates an opportunity for how the hopeful monster can find a mate. In any event, let's not digress back to that. So, when you have these breaks in between the centromere and the paracentromeric material, you can insert DNA. Now, where does this DNA come from? Well, there are retro transcripts, that is to say, take RNA and turn them into DNA, always in our system. But especially if you have a retroviral infection, you will have a lot of retro transcription activity, which will include not only the retrovirus, but it will also include whatever is being transcribed in the normal chromosome at the same time. So over here we have this gene being transcribed. There will be a piece of RNA, there will be an intron or something cleaved out, and that might get converted into a piece of DNA, which might get inserted into that section that was broken between the centromere and the heterochromatin. And so the centromeres have been shown to grow from the inside out. I mean, the growth area of the paracentromeric heterochromatin is at the interface with the centromere. In other words, the centromeres and the paracentromeric heterochromatin look like a tree ring. The oldest stuff is on the outside, and as you go towards the centromere, it gets younger and younger. And there's several papers written about this. This is not a hypothesis, this is actually published. Not widely known, appreciated, but it's published phenomenon. Now, it turns out that the other place where there are breaks and various exchanges occurring, it's called seeding and swapping, is at the interface between the euchromatin and the paracentromeric heterochromatin. There's several papers been published in this area, and so there's evolution going on at the beginning of the paracentromeric heterochromatin and at the end. And why is this relevant and important? Well, it turns out that if the adult female is changing, the fetus had better be changing too. These two things have to work together. The fetus has to change to match the adult, or vice versa. You have to have consistency between these two things. You cannot have a gigantic fetus and a tiny adult. Okay, these things have to work together. If it don't work, you will not get birth and you will not get a next generation. Okay, so these two things have to evolve together, and this goes back to why we don't have a simple phylotypic form. You have to converge. The fetus has to be converging at the same time that the adult is converging, because you converge to a phylotypic form. So I've actually preempted some of my discussion of evolution here, because you can see how the heterochromatin can evolve and how it could be used as generation-specific control keys. Now I'm looking at that. See if I can get a image up here, not upside down, but right side up. Okay, let's look at this for a moment. Here we have, for convenience, I picked mammals because they're nice graphics like this I stole out of someplace on the internet. And you'll notice that the bone structure is the same in all the mammals. As I say, the number of bones are all the same, and the articulation of them are all the same. What is different is their shapes and sizes. And it comes down to this. If you want the bone to be long and straight, basically you'll have the clones that make up those cells basically develop simultaneously on both sides, so it's straight, and if they develop rapidly, you'll have a nice long bone, and if for long term, you get a nice long bone. And on the other hand, if you don't develop that direction but develop the other direction, you get a wide bone. So the clones are basically developing different rates and for different periods of time. In other words, there's a differentiation phase, and then it's followed by a proliferation phase. And how long and how fast that proliferation goes on, I say how many generations it goes on, you will get different shapes. If you want a curved bone, you basically the clones on this side are more rapidly evolving, or not evolving, but proliferating than the ones on the inside, because the outside radius is longer. So the bone curves. If you want it to go straight, you want it to go wide, whatever. So what you're looking at here is different clones proliferating and differentiating at different times and for different periods of times, ultimately for the period of time. So from if you go down to the clone level, you have a differentiation process. Then you have proliferation. Then you have another differentiation, proliferation, etc. And so how long that clone proliferates will determine how big the clone is, how much space it occupies, etc., and how it interacts with all the other clones around it. And so you have these processes in sequence. Now, one of the problems that occurs in this hypothesis is if I've got thousands of different clones, and obviously I do, I've got almost that many different organs, tissue types and stuff. If you have all these thousands of different tissues and organs, can you really believe that the generation by generation each clone is going to be so well synchronized that you don't get some blob, you know? So how did I end up nice and relatively consistent here? Well, the answer is that we also have a master clock, pituitary gland. Okay, the master clock doesn't do anything except keep time. And every so often, it clicks over and it says it's time to send out a message, a hormone. So you get the puberty, bam, a lot of stuff have to happen in puberty. All sorts of body parts start changing in puberty. Boom, pituitary gland says it's time to be changed at puberty. Out comes the signal, and as long these different systems can be out of sync a little bit, but as long as they have the correct receptor available by the time that message goes out, that clone will receive that message and proceed as according to plan. And so they can be resynchronized by the pituitary gland, which will affect those clones that have a receptor and not affect those clones that don't have the receptor. So we do have a secondary mechanism which will synchronize and organize all these different clones doing all their different things. Okay. So I think that covers most of what I wanted to talk about today. I apologize, this is kind of rough. I had lost my voice. I've got things popping up on the computer screen that I didn't expect. But, I think that the essential message has gotten out here. I'm going to try to upload this to YouTube, let you go with that. Like I said, if you want details, I'm not giving you the details. I've got 30 pages of citations in this one little book, and this is a 100-page book, 30 pages of details. And there's some diagrams here that help tell the story. The last part of this book, other than the citations, has to do with the evolution, and that would be the subject of my next part of this presentation, how evolution occurs and why it occurs. Because the mechanism I've just discussed, the beauty of it, and one of the things that gives me faith that it might actually work, and the reason I'm actually talking about it as a hypothesis, worth talking about as a hypothesis, because it not only does it explain development, it just explains evolution. And I think, you know, to have to start off trying to explain development and finding out that you also have explained evolution is sort of psychic to me. But in any event, I did not start out here trying to explain evolution. I started out here trying to explain development. So have a great day. Like I say, look up my other videos. If you have criticism, I love criticism. You can find my email address on the internet. You can find my address on the internet. You can find me in phone books. If you have problems, if you want to criticize or have more information, feel free to contact me. Have a great day. Thank you.